Thank you all for coming to the last Conversation Series event for fall semester. I'm happy to announce the Conversation Series coming up next semester. We've got 3 great events, same time and place, and you're always welcome to show up for lunch anytime between 11:30 and noon. Coming up first in the spring will be Tyler DeRoche at the School of Sustainability with Charles Perrings and Ashley Kamhi, and Tyler will be talking about, are there any ecological conditions that are essential to sustainability? Next up is Ann Kinzig with Liz Barnes. And Ann will be raising the big question, what is the role of the university in solving society's problems? I think that'll be a great discussion. And last, in March, is a new professor that we're happy to welcome over to the Biology and Society Center. He's Zachary Horn, coming from the School of Social and Behavioral Sciences, and he has a background in both philosophy and psychology. And he'll be talking about the influence of cognitive biases on scientific explanation. So please join us for those. But for today, I'm really excited to welcome Jessica Bolker, who is here as part of a joint program event between the Life Science Ethics program run by Karen Ellison there. And if you're interested in that, I encourage you to go talk to her afterward. And just to give a little bit of background for those of you that are new to the conversation series today, the aim of the event is to break out of the mold of the normal specialized academic talk where people sit for 45 minutes and then ask a couple questions and then go on with their day. What we're really trying to do here is create a venue for interdisciplinary engagement around a big problem. And what I mean by big problem here is an issue that goes beyond any single discipline's ability to solve. And so part of the idea here is for everyone to bring your perspectives to the discussion, not just ask questions of the speaker, but really say, here's how I think we should tackle this, or here's an assumption that I think is problematic that if we think historically or philosophically or from a different part of biology, we can really open up new ideas about. So I encourage you to get involved, and the format will be that Jessica will talk for I think about 25-30 minutes, and then we'll open up the rest of the time for questions, and Sean will lead us off with that. Okay, so without further ado, Jessica, take it away.
Okay, I have been reminded to turn on the microphone. Is that okay, Jim? Okay. Yeah, I'm not going to talk for 30 minutes because I'm more interested in what you have to say than what I have to say. I think this is the official title from the poster, but what I was told when I was discussing with Beckett and others, what's the format, what am I supposed to do here, is that really what I'm supposed to do is say, what's a big problem? And the big problem I want to look at today, and that I'd like all of us, I hope, to look at from a lot of different angles, is the translational gap, of which I will give you a very simplistic definition. And the question that I've been struggling with is, how can philosophy of science, epistemology really, help potentially to bridge it, or to help us understand why that gap is there, and therefore what might be some useful approaches to trying to get across it. The particular example that I'm going to use, it's not the only one out there, to try and implement this approach is the question of animal models in translational research. So I'm going to give a couple of definitions in this talk that I know are simplistic. And we can argue about them. I'm fine with that. But that's only if that's something you guys want to argue about. So for my purposes, immediately, translational research, I'm using that as shorthand for the bench to bedside. So we have basic biological knowledge or research that is done with the intention and the hope and the expectation, ultimately, that we're going to be able to translate that into something that's clinically, practically useful, that's going to help people be healthier or be cured or live better lives. And again, a simple schema. There's more literature out there than I even want to think about trying to define translational research. Here's one version of a sort of loop that shows that we start with basic research at the bench, maybe not even an organism involved. We move from there to work in whole animals. And then we go through clinical trials with humans, and then eventually that's supposed to lead to bedside, that is to clinical treatments and applications. And ideally, we end up closing the loop, that some of what we learn from patients and how various therapies work in humans then informs what's the next basic research we need to do to move forward, to refine, to fix problems. The problem, as I said, is that translational research often doesn't work. And this disconnect is abbreviated as the translational gap. So we've got the scientist over here with the chemistry-looking flask and the test tube, and this unhappy-looking patient laid out on a table on the other side. And we have a cheerful skeleton in between and a rather rickety-looking bridge. So it sounds great in theory, but an awful lot of the time we don't get across from one to the other. And this is also sometimes referred to as the valley of death, where prospective, promising-looking therapies go to die. So why is it so hard to get across? One reason is that it's not simple. So the last slide showed you a very sort of cartoony, diagrammatic illustration, visualization, metaphor for this translational gap. This is a much more complicated picture. And the science and the ethics and the sociocultural issues that all get woven in here are, in fact, very complex. And there are lots of reasons it's hard. It's hard because disease biology is complicated and very often unknown. And if we don't really understand exactly what we're trying to fix or how it went wrong in the first place, that makes it more difficult to figure out how to fix it. Translational research is expensive. So resources are limited. You can't do everything. And that's another reason it's hard to get across. There are all kinds of issues related to the use of human subjects, which at some point in the process we need to do. You can't just realize that this drug doesn't kill mice and cures their cancer and then march into the clinic and start giving it to everybody. So human subjects issues and institutional structures. And this is more sociology of science, perhaps, than science, just by itself, if it ever can be just by itself. There are very different cultures between clinical research and basic lab research. People are trained in different ways, and it can be difficult to communicate or to share priorities or strategies between what are really two different subcultures in terms of the disciplines. The last piece, which is where I've tended to focus, is I think, and this is a claim or a hypothesis, that part of what's making it hard has to do with research practices, with how we go about trying to do translational research. And specifically, the role that this mouse to man, or animal studies over to humans, the role that this plays in the process, and the assumptions that are baked into that particular step. And there are a lot of assumptions I think we don't really think about as much as perhaps we should. So I've referred to model animals. And again, another simplistic definition, so we have one out there. What I mean in this case by a model is something that represents something else for purposes of studying it. So mice who are running through a maze might represent humans challenged with a particular behavioral task. This is the big one for translational research. Well, this in mice. I love that picture because it's a meta model. It's a model of Drosophila, and it was actually built like a Mr. Potato Head, so they could swap out different pieces of the phenotype to represent the effects of different mutations. So these are some of the classic models, but in translational research, we're mostly talking about rodents. Not only, but mostly. So here's how it's supposed to work. We do this basic research and deepen our understanding of the biology in a rodent, and then we can use that as a basis or a foundation to translate it up to a better understanding of the biology of human health and disease that is then going to allow us to make progress in the clinic. But as I said, it doesn't work all that well. Even when the science is fine, translation often fails. In fact, it fails something like 90% of the time, depending on what subfield you're looking at. People have pulled together data about this, and it's very depressing. There's sort of two categories of explanations for why. One is that the preclinical work, the work that's done on the bench in the animal models, just isn't good. That there's weaknesses at that level. And then the other category of explanations has to do with, maybe it's not what we're doing so much as how we're going about it. So it's not tactics, but strategy. So there's been a lot of attention lately, I told you I had a slide of this, to the tactical stuff, to problems with the basic science. It's being done sloppily, there are all kinds of pressures. There have been a couple of large-scale attempts to reproduce lots of basic science that has served as a foundation for the development of therapies, which don't work real well, and it's turned out to be impossible to replicate the original studies that those therapies were then based on. So that's a big problem right there. Because if this piece isn't solid, you can't use it to get to the next level. Now, these are, again, to oversimplify, essentially scientific problems. And we can tackle them with scientific tools. So what it takes to improve experimental design and analysis, that's about being careful and setting up your stats properly and doing your power analysis instead of saying, I've got 6 mice today, I'll do it with 6 mice. There are very well documented biases in reporting and publication of preclinical basic research. And there are strategies for dealing with those. For instance, having pre-registration of trials rather than do the work, see what you get, and then back calculate and say, that's what I was testing, which happens. And a certain amount of this has been formalized and by consortia and guidelines and so forth. One of the better known ones is this group called Camarades. So it's a collaborative approach to meta-analysis and review of animal data from experimental studies. And the idea is, people, could we just figure out what it takes to make this science solid and then have a set of guidelines? And you have to go through and say, yeah, I blinded this, and I've controlled for that bias, and I've randomized the other thing, and I did the power analysis, and I pre-registered my trial. And a number of journals, for instance, have said, if you're going to submit your basic research that's intended to be translated to us, we want you to follow these guidelines, which is great. What's not so great is last month there was a paper in Science surveying what effect that has had. So how many papers published since those guidelines were put out there and the journals said they were subscribing to them actually follow the guidelines, and the answer is almost none. So it sounds good, but we're not walking the walk yet on this. But that's not the only problem. Because even when the basic science, the work that's been done at the bench in the animal model, even when that's perfectly solid, it still often doesn't translate. And my argument is we need to step back from just thinking about, did we do the science right, to the epistemology. How are we doing the science? And that opens up cans of worms, various sorts. We need to think about the different kinds of validity that we're claiming for preclinical work. We need to think about the context. What was the question, and what's going to count as an answer to that question? And how might the way we go about doing the research shape the science, the direction it takes, the kinds of answers that are available or perhaps less accessible using particular approaches. So again, this is where my focus has been, looking at this particular part of the process. So there's a fairly small number of animal models that are dominant in biomedical research. This is an old screenshot of NIH's page of sort of pre-approved models. If you want to apply for money from NIH and you're not using one of these, you better have a really good justification, because these are the ones they've decided work. And it's not they, I mean, it's us, it's peer review. So these are the canonical models, sometimes called. I love the religious overtone of that. That said, some are more equal than others, and this is who's more equal than others. So it used to be rats were much more equal than anybody else, and once we gained the ability to do powerful genetic manipulations in mice have been taken over. So this is an article by some locals. Thank you very much. I love it when people do this kind of work, because I want to know this stuff and I don't want to do this work. It's too hard. What this shows is sort of the dominance of these particular models. Now, the take home in the article was, you know, just because NIH canonized these 13 species doesn't mean people aren't using others. In fact, people are using a broader and broader diversity. And there's been gradually increasing use of more and different species since that web page was put up. That said, they had to completely rescale the figure to show that, because these are the mice and the rats, and everybody else at this scale looks like a flat line. I mean, in the rest of the figure, in the other parts of the figure, you can see that when you rescale it and take off the mice and the rats, there is a lovely increase showing more and more diversity of models being used. But it's still swamped by these dominant models. And I think that's a problem. And I think, in fact, that the dominance itself is part of the problem. And I don't know what the solution is, but I'm pretty confident that the first thing we need to do is recognize the problem and work on articulating some of the specific obstacles that the dominance of rodent models poses, and then we can get going on figuring out what to do about them. So this is from the paper that I know a bunch of you guys have read already. Thank you. So it's a list. So dominant models can get in the way, can obstruct translation if they embody one theory of disease but don't actually make it possible to explore any others. There are always going to be species disparities, but if you've only got one model, then you can't get around them or compare them. Lack of context, lack of variation, that comes partly from the industrialization of mouse colonies, rat colonies, inbred strains, and so forth. And ultimately, in some fields, it seems like the model has actually become what it's all about. So what you publish is improved treatment of an induced disease state in a mouse, not, well, this is actually useful for people. And induced disease state improvement measured with some very standardized, say, behavioral task in a mouse. But that's what becomes publishable. And it loses track of this was supposed to be translational, but we're not actually getting to assess it at that level. So I'm better at problems than solutions. A few of the solutions that I was able to come up with, and I'm hoping and expecting to hear a lot more in the discussion. As I said, first we have to recognize the limitations as well as the strengths of the currently dominant models. We need to clarify the criteria up front for what counts as a good model, why and how do we choose a model, and we need more different ones. We need to diversify the portfolio so we're not stuck with just one biological representation or perspective on a given disease or problem. And we need to add us as another species in that portfolio. For an awful lot of diseases, we don't know the biology of the human form of the problem well enough to know how well we're representing it in some other species, because we don't really know what the target is. If you're not clear about what the target is, it's really hard to hit the target. So in terms of epistemological strategies, sort of thinking about how we're going to get this knowledge, we need to go at this from all kinds of different angles. I mean, certainly, we need to use the best possible science. We need to actually follow the guidelines and do things right and not take shortcuts and make sure things, do what we can to ensure that things are going to be reproducible and so forth. So the science has to be good. But there are other places to look for tools. So we can look to epistemology, we can look to ethics, we can look to sort of sociology of science to understand where some of these challenges are coming from and how we might begin to deal with some of them. So I'm better at questions than answers, so here come some questions. Some of the questions I think we need to be asking up front are, what is it that we really need to learn to enable translation? So why are we studying animal models other than because we always have? And what can they tell us, but what also can't they tell us? Once we have decided where we're headed or where we want to get to, what research models and strategies will help us get there? And how are we choosing them? And then sort of going meta, how can we clarify the questions and the answers and the assumptions related to model use? Because this is absolutely standard operating procedure in basic biomedical research. And one of the effects of that is people kind of stop thinking about it because it's how we do it. It's how it's done. And I think we need to take a look at what the implications of how it's done might be for what we can get out of it. So one approach is to think about it sort of like a philosopher. So start with, what's a big epistemological issue here? How does research strategy shape science? That's not a science question. It's not a question anybody in my Department of Biological Sciences would recognize as a legitimate thing to be asking. But we can then get from there, if we start there, we can then think about, what does that look like in a particular domain? For example, translational research. So in this domain, how does the dominance of core animal models maybe obstruct translation to humans? That, it turns out, is publishable. And if you think through to that level, then you start to have a research agenda. Because that then leads you to, okay, what do we need to know to fix this? What do we need to do differently? What do we need data about? What changes in business as usual with model choice and model use might actually help bridge the translational gap? That's the end of what I have to say.
So thanks so much for the discussion, Jessica. And so my name is Sean Cohmer. I'm a PhD candidate here in the School of Life Sciences, Center for Biology and Society. And I study the history and philosophy of science, particularly the history of animal choice in biology, starting in the late 19th century. So I'll just kick off the discussion with some of what I'm interested in, and that is animal choice. So this also ties into some of the interests in the department. So there's this recent advent of a methodology in science, CRISPR-Cas9. And so a lot of people who I've been talking to at biology conferences have said things like, okay, now we have CRISPR-Cas9. The vista of animals we can work with in the laboratory has broadened significantly. We have this new methodology. Now we can use all kinds of different organisms. But the rationale for choosing the organisms has been lost on a lot of scientists because it's sort of a generation gone by. We've got institutional and practice-based standardization of animal models in the laboratory. So what in your mind constitutes a good rationale for choosing animal models, given your experience in writing about this?
I guess I'd start with being super clear about what the question is you're trying to answer. And then you need to look for an animal model that represents or embodies whatever it is about biology that needs to be there to enable you to answer that specific question. And there's a balance always between something that's manageable, that's tractable, and something that's more or the best possible representation. So, the best representation of humans is us, but there are lots of logistical reasons we can't just use us. CRISPR is really interesting because it adds a dimension of tractability to just about anything if what you want to study is swapping genes in and out. But that might not be the only thing you want to study.
I agree with most of what you said in your statement here in your paper, but there are additional things that have to be added. For instance, I think we have to keep in mind that most, or certainly a lot, of mouse disease research is not designed for the purpose of translation. It's also knowledge-based or theoretical. We're trying to get at theoretical questions and ideas, in which case the inbred strains of mice are very useful. You're focusing on something specific, the genes involved, et cetera. The environment, your environmental point is an excellent one. The mice are kept in the research colonies here that are they're not exposed to all of the environmental pathogens that they're exposed to, for instance, in the wild, and yet that is a rule and regulation of the NIH and people like that. We try to get around that by sometimes enriching the colonies and handling the animals ourselves so that we don't introduce unnecessary stresses or things like that. So I think another thing that you touched upon, which has to be developed, is that in terms of the translational gap, there's a lot of bias that has to be overcome, both in terms of the NIH or the NSF study sections, which focus in a certain direction, as you mentioned, but also in terms of the translatability of research between the researchers, basic researchers, and the clinicians. Very often you'll get the statement that, well, this is a mouse model, therefore it's not useful for human disease. You said the opposite in your paper, saying that this is the essence of translatable research. Mouse models are very important. But it goes the other way too. You're going to see a lot of reviewers saying this is a mouse model, and it doesn't have much implication for human disease. Well, I think you have to understand what the limits are, which I think is also what you're trying to get at. And then my final point is, I think one of the things that has to be incorporated, because biology of disease is so complex, you have to take into account environmental factors. There's a myriad of individual molecules that are involved that have different effects at different times, different stages of development, et cetera, et cetera. I think we have to start incorporating computers to allow us to understand what this complexity of interaction is all about. And then finally, related to this, I think this is why they're getting to personalized medicine, so that, okay, an inbred strain of mouse is not equivalent to the human situation, which is outbred and a lot of environmental factors. But again, why one treatment doesn't work for a particular individual, but it does for another, is also why you use inbred strains of mice. That could give you information as to why it works in one human but not another. But I think we have to develop those ideas and truly understand the complexity involved. So those are just a couple of comments.
Thank you. I will say that certainly a great deal of work on mice is about basic understanding. That said, the mission of NIH and the ultimate rationale for NIH to support research is not healthier mice. You know,
But it is knowledge.
it is knowledge, but big picture, it's not the National Institutes of Knowledge. You know, there's an overarching piece there.
You need the knowledge in order to develop the translation research.
Yep.
I think we'd be better off if we stopped calling organisms models and thought about actual models as models. So to say a mouse model, it's a mouse, and a bunch of them is a mice. And because when we call it a mouse model, it obscures the fact that what we're doing is we're studying mice, we're developing conceptual or mathematical models, hopefully, from the research the knowledge we gain from mice, and then we're trying to apply those to other organisms. We wouldn't be doing this if we weren't trying to make humans healthier. I totally agree. The best model for humans, the best model is a bunch of clones of me, frankly, if we're talking about health. But I get that other people want to be healthy too. So the best models are the models that are going to represent the populations we want to cure. Totally agree. But the problem is, in the 1900s, the guy Crow, who won the Nobel Prize, said for every question there's a perfect organism. It's total bullshit. For every organism, there are interesting questions that are relevant. Right now, the organisms are humans. The questions are health. So then the question is, if we can't mess with people, what can we do to build theoretical models that would translate to people, right? And I think the idea that they have Daphne up there is ludicrous over like another mammal. I mean, it's going to be something that hasn't been separated by humans by very much evolutionary change, right? And mice have got to be tens of millions of years separated from humans.
90.
So what's that?
90.
90, yeah. Tens of millions. So these are all horrible models, but so what's the best model we could get, right? I think this is philosophy of science. Like most people don't get this in undergrad education when they learn model organisms. Why don't we teach them about models? And then ask, well, what organisms do you think? I bet an undergrad could come up with a better model if we approach it that way than the models that are on that board.
Can I add something?
Yeah, sure.
So there's actually a really interesting paper that came out a few years ago where a group looked at essentially a phylogeny of genes known to be associated with human diseases and how far back they go. And some of them go very deep. And those are things that might actually be possible to learn about from studying Daphnia. And others only pop up kind of post-mammals or post-primates. So I think we don't necessarily always need the closest thing. It comes back to what exactly is the question, and that's got to drive what's the best model.
Yeah, but the minute we stop saying model organisms, we can start to have that conversation you just said, because we know there isn't a model organism. There are just organisms which you can use to build models about people, right? But I think there's a philosophy of science that's being obscured, right, by the term model organism, as if there is a model organism.
I think model organism is more of a sociocultural phenomenon than a philosophical one, honestly.
So I had a question. When you went through your strategies, you used the term we more than once. We should do this. We should think about that. And I wonder, who is we? Because I don't work in a lab with animal models. Like, do I get to be a part of the we who gets to decide? Or does the we, I mean, I imagine maybe the we should be the NIH, given that they're the ones funding this. And so if they're not part of the we, then the funding doesn't work and people won't do it. Like, who is the we that we need to convince that they need to think about this?
So that's a really good question. And I'm never sure if I should say we or say you guys. I mean, I don't study mice. The reason I use we is I don't think this is something that can be fixed from the outside. I think ultimately it's going to have to be people who are involved in doing the research, who have a stake in the outcome of the research, who are the only ones who can effect any of the changes. And that is actually one of the reasons that having some models be so dominant is problematic. Because if you work on some weird little one-off model organism or species, and you put in your grant proposal, almost everybody on that review panel probably works on one of the biggies. And they've bought into that, and they see the strengths of that, and they're there because, and they're successful because they're convinced and have evidence that working on rats is the way to go. So there's a sort of inertia to it. NIH is moving some now, at least on the reproducibility, how solid is the science stuff. I mean, nobody ever asked me to sit on any NIH panels, but I do talk to colleagues who do. And they have new review criteria that are very specifically focused on that and on experimental design, having recognized that there's a problem and we, that is the community doing the work, need to fix it.
So this is lovely, and I'd just like to point out that one of the main themes in evolutionary medicine is path dependence and how bodies are suboptimal because developmental pathways get fixed and they can't be transformed in any big way. And I think the problem you're calling attention to here is another one of path dependence, of people using certain animal models and making big investments in ways to manage them and cages and genetics and all the rest. And once you get down that pathway, it's very expensive to do anything else. Just a comment that fits in with your developmental biology approach to all these things.
So if I understand what Mike was getting at earlier is that, well, if you call it a model organism, then there's several easy ways to misunderstand what you might mean there. One is that it's an exemplary organism. So that's one sense of model, right? And there's one that you might also think that, oh, well, if it's a model organism, that it could be a model for anything, and it's as if being a model were somehow intrinsic rather than relative to the question. So part of what I took you to be doing there is to say, like, well, what if we change the phrasing, and is that going to sort of push people in a different direction cognitively? When we say, like, well, let's talk about the species, and then talk about the questions that the species is appropriate for, instead of trying to label certain things as models, and then sort of making it easy for people to get entrenched into thinking that this is a general model, rather than something that can be used to answer a specific question, or maybe several questions. To pick up on that, I want to think about what are broader strategies that we can use and that maybe history and philosophy can contribute to in this context of pushing people to think differently. So I think one of the issues that you were raising was we don't have clear representations for the community, either narrowly or broadly construed, of when things have worked or failed, and how funding bodies like the NIH are driving decisions, and whether those have anything to do with the track records of the experiments and species for specific questions. So I think one example there was the research you showed of just trying to get to grips with how funding is changing the amount of research being done. But what are other things that we could do that would maybe help intervene on the panel decisions or on the community's broader awareness about when certain species have worked well for a question, or when certain questions have been asked and strategies have failed in addressing them? I'll sort of toss that out there, and maybe you can have a thought or two to get it started. It's like, where would you see a useful pressure point be to intervene on how the broader community evaluates specific species with respect to specific questions?
I think it's super hard, because the community is the people who have been productive and successful because they are, getting publishable, fundable, good results out of the work they're doing, as judged by their peers who use similar kinds of systems and approaches. So it's really hard to say, by the way, this is not working. I think that there's some very important work to be done from the history side here. And I occasionally try and pick my historian colleagues' brains about this. Where have there been historically places where we arrived at a wrong conclusion or something was written off because it was the wrong organism. Or it was weird in ways that were relevant to the question that we were trying to answer, and therefore we got the wrong answer. And I know of a couple of those, but other people know more of them. And I think that that would actually be useful to look at because maybe there are some patterns. You know, how do people realize that what they're doing isn't working? And what do they then do about it?
Yeah, so I'm a little fuzzy on what it is that we're trying to fix. So Mike says, why don't we just talk about organisms? But there's this thing about being a model, and we're not really talking about modelness. And so you said philosophers of science can help. Jason Robert's written a lot about this topic, but you know, we need to work more on what it means to be a model and why we have them and what work being a model does. So Eric Davidson convinced NIH to think that sea urchins should be a model for gene regulatory networks because he sold the idea that gene regulatory networks are really cool and important and will help us understand ourselves, but also biology and everything else that we care about. And he bought into the idea, we'll call it a model, because that will be what convinces NIH. So there's this category, this box of modelness that is doing a lot of work, and as Jason has shown and you have shown and others, some bad work. So it seems like the problem lies in the what do we mean by model and what do we think it's doing, and if we need to have them fix something, it's sort of an understanding of what can be modeled, how can it be modeled, who can do the modeling, what you gain and lose by thinking in terms of models, and that sort of thing, right? So we're talking about organisms, but the modelness seems to be important.
Mm-hmm. So, Christian.
They're running the show. I don't get
Oh,
to
I
say.
have a totally different question. That's why. In the slide where you showed only 10% of this translation happens, my instinct is the same, that seems low. But how do we know that that's not just the rate at which basic research can be turned into clinical applications? So even though, again, my instinct is we could probably do better, but how do we know that? Because that's just me intuiting not being a researcher. Maybe we can have a better return on investment. Because if you're going to try to change things to try to increase that 10%, that costs resources that are scarce, that are limited already. So you would want to have confidence that changing things and using those resources in that way is going to actually improve that in some way. And I don't know how you go about making that judgment or how you would know that this is an effective thing, that we're trying to change the right thing if this is the right problem to fix the translation problem, if that makes sense.
So can I say something to that? So the 10% is research that's been done in animal models that has been sufficiently promising and checked all the boxes that it has moved into clinical trials and then not panned out. So for me, the question is, what are we missing? What additional criteria or filters do we need earlier in the process to do better than that? So 90% of the stuff we have every reason to think will work doesn't work. That's a problem. Because, as you say, it's expensive.
Not to disrupt the conversation, but perhaps to expand it, let me suggest some other examples that don't deal with human health and human subjects. And my particular interest is fire. For a century now, the U.S. has studied wildland fire with the explicit purpose of applying it. And earlier this year, National Academy of Science had a workshop. Forest Service chief and chief scientist got up and said, science applies the solutions? Why don't we have more results in the field? So we're missing something in the science. And I look at it as a historian and I say the science has never had the answers. And one of the issues is that we use the capability of the science to define the problem. And we want to do that in part so we don't have to deal with values and politics and all these other things. In fact, they are social and political problems. And they're not scientific questions. So repeatedly, the science has not only failed to make the translation, but it has been used to inform really poor policies. Almost all of our bad choices have had the science of their day behind them. So there's an effort, I look in vain to see where science has made a practical difference in the field. And in some ways, we would have been better off with traditional. We would have been better off studying what Native Americans had done for thousands of years because it was empirically based. The science obliterated that, came in with models. The models have repeatedly failed. So I think part of it is it's an issue, how is the problem even defined? And what can the science do? There may be stuff it just inherently can't do it.
I'm happy to have other people do more of the talking since there are other people who want to talk.
Yeah, just to follow up on those two questions, you can broaden the field and look at the general translation of basic research into what is now called development or technology in engineering and physics and everything else, and you come up with pretty much the same answers, that only about 10% gets translated into some usable product. And even in business, what do they say, 80% of all new businesses fail. So, but I wanted to throw something else into the mix that I was thinking that, only looking from the outside, that one of the fundamental problems in translational biology is the whole system of research and funding, because there's so much pressure on scientists, on researchers, to publish, to do research, to get the research done, to get a new grant, to finish this grant so you can write another grant, and to have publishable results. And so everybody's being pressured to work as fast as they possibly can and keep getting more money and keep the lab funded and so on. And as a result, they're being pushed into publishing things that probably shouldn't be published yet. They should better wait another year or something and do another year of research or 3 years. But they can't because the pressure, the institutional pressure is there on them. So maybe you can respond to that.
I don't know there's much to say other than I think you're absolutely
those
right.
in the field, I'm just doing this from the outside, but for those of you who are in the field, do you feel that's part of the problem?
Yeah, I definitely think you've hit on a lot of important issues. And I think you do have to get to the NIH or the NSF. I'm thinking NIH primarily because that's what you're talking about. NIH study sections and how they do funding. And I think there's a lot of bias in NIH study sections because of the way they're set up. And I think the study sections were great for 30 years ago when there was more money than they knew what to do with, but the money is so scarce now, it's an archaic system. And so they're pushing in a certain direction, and you're not getting creative and novel ideas that may be useful for translation. So I think you are touching on very important issues.
Just to jump in there for a second, I think we've touched on two of the different obstacles that Jessica brought up. And so one of them I could maybe cast as, and this is sort of picking up on Steve's concerns, have we set up animal model research in such a way that we would actually know when we're failing, right? So one of the issues is that, oh, well, we started doing this and it worked a couple times. Let's definitely put in another $1 billion, right? And you kind of build in the entrenchment cycle there, where, well, we've always been doing this this way, and it works sometimes, and the best thing we can do is keep pushing forward with it. And you never sort of step back and say, well, should I be trying something radically different? right? And ideally, if we can be empirically minded about this, we should be keeping track of whether we're failing so often that we want to seriously consider alternatives. And that kind of gets back to this issue of what is the track record of different animal models. But I think there's also another concern, and what I heard Steve getting at, which is the biology too complicated for the strategy, right? So maybe the fires and the forests that Native Americans were sort of working within were best handled in a way that doesn't fit within contemporary modern science. And that's why we would be better off if we hadn't had the last, 100, 150 years of forest management science, right? Because, in fact, the way that science has been constructed is significant or consistently misleading with respect to certain complex phenomena. So that would sort of be an ontological question of, well, maybe disease or maybe organismal development is so complicated that the way that we've set up existing research and the kind of underlying ideas of translational medicine based on animal models is misbegotten from the start. So there may be two different issues there, and it would be nice if we could try to actually disentangle them going forward, not just like here in the conversation, but more broadly, because they would really have, one of them you can fix, right? One of them you can say, oh, I'm failing so often, I want to try something different. The other, you have to say, yeah, I need something radically different then. If it's too complicated for my current strategies of science to work, we need a different kind of science.
I was going to go back a while to what Beckett asked earlier about potential things to do to even, I guess, determine the scope of the problem. Sometimes in conversations like this, I start to feel like really depressed, like we've gone backwards and there is no progress and it's just all bad. But I think in all actuality, we actually have probably produced quite a lot of good science in the last 30 years, and that maybe some of the problem is wrapped up in the inability to access large data sets, or the inability to take large data sets and do any sort of analysis on them. And I wonder if systems like, so there's clinicaltrials.gov for when clinical trials are going through, if you couldn't do a similar sort of system with preclinical trials to kind of have some sort of accountability, but also sort of a tally of what people are doing and what their results are in a centrally located place that allows for a little bit easier sort of assessment. But also thinking through like public accessibility of science and the fact that journals and articles and publications sometimes can be very difficult to access. And so it's difficult for someone who's maybe involved in computer science or is someone who could run a lot of these like large scale analyses aren't having access to those large sets of publications.
Just going back to the complexity issue, I think we do have to think in terms of, right now, all of our systems, all of our research are geared to simplicity, to reducing it to the simplest factors possible. And we do have to start thinking in terms of, how to start complexifying everything. And that's where I think you do need to use computer models in conjunction with the animal model, I think you have to start advancing the complexity research using computerized systems.
So I was thinking about Jane's question of what is a model, and I was thinking of an earlier claim. Someone said, I think it was you, that sometimes we use model organisms to learn about some specific aspect of human biology, but sometimes it's just to generate theoretical, more general biological knowledge. In the first case, it seems clear that the model organism, I got scare quoted now, is a model for some aspect of human biology, and it seems straightforward. In the second case, I mean, it's almost like the organism is a model for life in general, but it can't be that because there's too much variation in biology. So it's a model for something with sort of cloudy borders, and it's an empirical question itself what organism is a model for when you're using it to generate just general knowledge, I think.
Can I respond to that quickly? So this is, I think, a really key point, which is we have to say, what do I mean by model? What do I think it's doing? And I actually published a paper some years back where I said, look, there's at least two different ways we use models. There's two different things we mean. One thing that we might mean is a proxy. So mouse representing human, which is quite targeted. And the other would be what I called an exemplary model, sort of mouse representing animals, or Drosophila for studying genetics. And it turns out if you think through these two different, really quite separate uses of model, you end up with a different list of criteria for what would be a good model. So it really matters to not just have a big box called models.
I was actually just going to plug that paper of yours.
Thank you. I should have let you do it.
Models of and models for. So just a couple of quick thoughts. One of them goes back to the question that we just had from Alexis. So one thing Jessica did do in the talk, however briefly, was mention that there is a movement afoot to register preclinical research. And it's an interesting starting point. It actually responds to two different kinds of issues. One of them is the issue of actually collecting everything together in a place where it's centrally accessible. But the bigger problem that it actually addresses is the publication bias issue, this idea that we have got a gazillion labs doing a gazillion things and publishing a tiny fraction of the things that work and nothing else. And so as a result, we see a massive, well, at least the prospect for a massive duplication, not replication, but duplication of efforts that are just as bad as the other efforts were that aren't going to get us anywhere. I'm disinclined to be at all optimistic. I think Alexis's optimism is adorable, and I'm going to chalk it up to, I don't know, youth or something. The older I get, the more concerned I am that what we're basically doing is wasting billions upon billions upon billions of dollars a year on stuff that generates some publications and nothing else at all.
So to kind of follow up with that, as a room full of scientists, researchers, I'm interested in learning what you guys think of, or if you think at all of, the in vitro models, such as organs on chips, bioprinting, 3D bioprinting of organs nowadays, and the fact that they are getting more and more complex and they're utilizing microfluidic chips. So they are becoming more systemic. How do you see that playing into your research and working towards those 3 R's of refining, reducing, and replacing animal models?
I have a quick response. It's cynical.
Really?
So it goes back to the point Randy made about pathway dependency, right? I mean, we are so far entrenched in laboratory practice in particular ways that the introduction of new tools and techniques is nice, and maybe at some point in 100 years or so, we'll actually see the widespread adoption of these kinds of techniques. But right now, it's so hard, even for an experimental scientist, to move from one animal to another, to follow the question where the question goes, from one animal to another, is dangerous career-ending kind of move, because if you do that, then you're going to have to begin building up your laboratory around a different kind of animal with all the different techniques that are required and so on. There's going to be a delay. You're not going to get tenured, and then you're just going to, be done. It could be the case that scientists, as they get older, could move around from animal to animal, except that empirically they don't. And so even though we see some beautiful new techniques emerging that could enable us to have a much better representation of the actual biological world that we're interested in studying, I don't see that we're going to see widespread adoption until everybody dies.
I think James, I'll have a comment.
So could we come back to Sean, please? So Sean, with your Morganisms, so T.H. Morgan won a Nobel Prize for working on fruit flies, and that's what we know. But tell us about Morgan briefly to reassure us that you can study lots of organisms and it's okay. Or you could.
All right, so the final word from Sean.
Yeah.
So yeah, I did this project on T.H. Morgan and Morganism. So the lesson from the late 19th century and the sort of transition from natural history approaches to doing biology to experiments in the laboratory taught us that experimentation with a lot of different organisms was really hard to do and could be done. But it also taught us, as you sort of transition into the 20th century, we have this comparative biology that's going on in the late 19th century, and the transition is towards a more, Kelly mentioned this sort of unity of life concept. And I've been trying to trace back this notion historically of the word animal model in history. And the earliest instance that I see the words animal and model together are used in this instance in Ludwig von Bertalanffy's book, Problems of Life. And what he does is he's trying to generate a sort of abstraction from the specific cases of biology, of organismal biology, to a more general conception of like what counts as life, what are the unifying processes that underlie what we understand as life. And this is what we see going later into the 20th century, and you know, this is the sort of anti-case of Morganisms. But there is a rationale for choosing organisms in the late 19th century, and I think we do well to look back to the history and think, how were people like Thomas Hunt Morgan choosing animals in that day, and sort of try to replicate some of those efforts.
So thank you all for coming. We are now out of time, although the conversation can continue. Thank you to Jessica for getting us started with a wonderful discussion.